So today I'll be discussing about Maxam Gilbert DNA sequencing, and it is the art of reading DNA through chemical clues. So let us imagine you are an archaeologist who discovers an ancient scroll written in a secret code. Unfortunately, the scroll is too delicate to unfold completely. Instead, you carefully tear it at specific letters and examine the resulting fragments. By studying the sizes of these fragments, you gradually reconstruct the original message. This detective-like approach is exactly what scientists used in Maxam Gilbert DNA sequencing, one of the earliest methods developed for reading the sequence of DNA and developed in 1977 by Alan Maxam and Walter Gilbert. This technique marked a turning point in molecular biology. For the first time, scientists could determine the exact order of nucleotides, the A's, T's, G's, and C's that make up the genetic code. So, why was sequencing needed? So, every living organism carries genetic information encoded in DNA, and the sequence of nucleotide acts like a biological instruction manual, determining every thing from eye color to cellular functions. So, scientists knew DNA was composed of four bases, namely adenine, thymine, guanine, and cytosine. But knowing the components was not enough, and the real challenge was how do we determine the exact order of these bases? So, reading DNA was like reading like it was like trying to read a book without being able to see the letters. So Maxam and Gilbert developed a clever solution based on chemistry, and the fundamental idea was like unlike modern method that copy DNA, Maxam Gilbert sequencing directly analyzes the original DNA molecule. So the principle is very simple: break DNA at specific bases and use the fragment size to determine the sequence. Scientists selectively cut DNA wherever a particular nucleotide occurs. So by examining the resulting fragments, they can identify the position of each base. It is a similar to solving a puzzle by studying pieces of different sizes. So the steps that is involved is first step is labeling of the DNA. The first step is to label one end of the DNA molecule. Scientists attach a radioactive phosphate group to one end of the DNA strand. And now you may ask why? Because after fragmentation, only fragments containing the labeled end will be visible. Think of attaching a glowing light to one end of a rope before cutting it into pieces. No matter how many pieces are produced, you can always identify the pieces connected to the glowing end. This radioactive labeling was essential for detecting DNA fragments after electrophoresis. And another step is dividing DNA into four reactions. The labeled DNA is divided into four separate tubes. Each tube contains chemicals designed to react with specific bases. The reactions are carefully controlled so that on average each DNA molecule cut only once. This produces a collection of fragments of different lengths. The four reactions are G reaction, cut primarily at guanine, A plus G reaction, cuts at both adenine and guanine, C reaction, which cuts primarily at cytosine, and C plus T reaction, cuts at cytosine and thymine. These reactions create unique fragmentation patterns. And step 3 is chemical cleavage. This is the heart of the technique. Special chemicals modify particular DNA bases. After modification, another chemical treatment causes DNA backbone to break at those modified positions. Imagine marking specific letters in a sentence with invisible ink and then applying a reagent that tears the paper precisely at those marked letters. The DNA is essentially transformed into the collection of fragments whose length correspond to the nucleotide position. So next step is electrophoresis, and now scientists have thousands of DNA fragments of varying lengths. So next challenge is separating them. This is achieved through polyacrylamide gel electrophoresis. DNA fragments are loaded into a gel when an electric field is applied. And smaller fragments move faster, as you all know, and larger fragments move slower. A gel acts like a molecular sieve. A useful analogy is children running through a crowded playground. Small children weave through quickly, larger children take longer. Similarly, shorter DNA fragments travel farther in the gel. And another step is autoradiography. Since the DNA is radioactively labeled, scientists can visualize the fragment using X-ray film. The gel is placed against photographic film, and radioactive emissions expose the film and create dark bands. This process is known as autoradiography. The resulting image resembles a barcode made of DNA fragments. Each band represents fragments ending at a specific nucleotide position. And next step is reading the sequence. This is the exciting part. And scientists examine the pattern of the bands across the four lanes. The sequence is read from bottom upward. And why is this? Isn't it? So, because the smallest fragment travel the farthest and appear at the bottom, each successive band reveals the next nucleotide in the sequence. For example, lane band G present, A plus G present, C absent, C plus T absent. This indicates a G at that position. By moving upward band by band, the entire DNA sequencing can be reconstructed. It is like reading a ladder rung by rung. Why was this method revolutionary? Was because before DNA sequencing, genes were largely mysterious. Scientists knew genes existed but could not directly read them. Maxam Gilbert sequencing changed that. For the first time, DNA sequences could be determined experimentally. Mutations could be identified. Genes could be analyzed directly. Molecular biology entered a new era. This method helped lay the foundation for the modern genetics and biotechnology. However, it had many limitations, like, for example, despite its brilliance, Maxam Gilbert sequencing had drawbacks due to the use of hazardous chemicals. Several reagents used were toxic and difficult to handle. Radioactive materials, the technique required radioactive labeling, creating safety concerns. And another is technically complex. Multiple chemical treatments increase the chances of errors. Another is labor intensive. The process required considerable manual effort and limited scalability. That means sequencing long DNA region was quite difficult. So, why was it replaced? Around the same time, another technique emerged, Sanger sequencing. Sanger sequencing proved safer, simpler, more reliable, easy to automate. So, as technology advanced, Sanger sequencing became the dominant method and eventually paved the way to modern generation sequencing technologies. Today, Maxam Gilbert sequencing is rarely used outside the educational and historical context. So, the legacy of Maxam Gilbert sequencing, although largely obsolete today, Maxam Gilbert sequencing remains a landmark achievement in science. The greatest contribution was demonstrating that DNA, the very blueprint of life, could be read directly through clever chemical analysis. It transformed genetics from a field of speculation into one of the direct observation. The method reminds us that scientific breakthroughs often come from asking simple but profound questions. If DNA contains life's instructions, can we learn to read them? Maxam and Gilbert answered that question with chemistry, ingenuity, and a method that opened the door to modern genomic revolution that shapes the modern biology today. Thank you.